Hello, this is class Bio 225, Genetics from Genes to Genomes, Chapter 5, Part 2, and this is Dr. Lucy Abgarian. During last lecture, we talked about DNA recombination. Today, we will go a little farther and see how the recombination plays role in DNA mapping. Okay, so now let's talk about mapping genes by comparisons of two-point crosses. So here we will look at the gene fragments from left to right orientation, and that is arbitrary for mapping purposes. We can look either way. However, we are going to look from left to right now. It is known that the most accurate maps are obtained by summing many small interval pieces rather than doing the whole big pieces of gene mapping. So here on the graph we have gene pairs here, and we have the recombinant frequencies about which we talked during our last lecture. And now let's look and prove what I have said before about accuracy of smaller pieces of gene mapping. So here we have YW and WM. So when we look at that whole gene YM, we see that YM is 34.3. However, when we sum up YW and WM, the sum of these two is going to be 33.9, which is more accurate than the 34.3 data given. And the same situation goes for case C, where we have YW, WV, VM, and MR. So the summing of these small pieces, which is given in D, will be 55. However, when we measure those pieces separately, YW is 1.1, YV is 33, YV, which is 33, YM, which is 34.3, and YR, which is 42.9 by direct measurements. So from this, we have to remember that smaller pieces are more accurate summing up the small pieces than looking at the bigger pieces of gene maps. Two-point crosses have some limitations, which include the difficulty to determine gene order when these two genes are too close to each other, and also the knowledge of the actual distance between genes, which we kind of saw already in the previous example, that the sum of the gene fragments is not always adding up to the same as the whole gene looking simultaneously. Also, which we will observe this during our next slide with an example, that the pairwise crosses are very much time consuming and also very labor consuming processes. So now let's talk about three-point crosses, which provide faster and more accurate mapping. Okay, so now let's look at the test scores of triple heterozygous phenotype 1. So we have parental line female VGBPR, VGBPR, which is crossing with male VG plus B plus PR plus, VG plus B plus PR plus. Here, some things to note, that plus is dominant gene, and VG segregates together even though V and G are separate genes. B segregates separately, and then PR segregate together, even though here we are dealing with 5 separate genes: V, G, B, P, and R. So now, because of the simultaneous segregation, the phenotype 1 will be all identical all the phenotype type 1 will be identical, and it will be VG, B, PR, and VG plus, B plus, PR plus. I think this should be clear that this is due to simultaneous segregation. So now let's do a test cross of VG, BPR, VG plus, B plus, PR plus with male VG, BPR, VG, BPR, and look at what will happen. So test score progeny would be as following. We will get the most of the progenies will be similar to parental combination for all three genes. So the most will be VG-BPR, VG plus, B plus, PR plus. So ideally, it would be a common sense to think that all of them were supposed to be parental combination. However, we see a smaller population of different combinations, which is due to recombination taking place. So, and we see some population of VG plus BPR and VG B plus PR plus, which is a recombination for VG relative to parental combinations for B and PR. And then we are seeing even smaller population of VG plus B PR plus and VG B plus PR, and this is recombinations for B relative to parental combinations for VG and PR. And the least amount with the following gene progenies we are seeing as of VGB PR plus and VG plus B plus PR, which are the recombinations for PR relative to parental combinations for VG and B. So let's analyze the results of 3-point cross. So test cross progeny have 4 sets of reciprocal pairs of genotypes. The ones that are most frequent pairs, they have parental configuration of alleles, which we have seen already in the previous slide. And the least frequent pair are from the double crossover. In this case, it's the one that gives the 13 and 9. So examination of double crossover class reveals which gene is in the middle. So you are having one flipping and then another flipping over it. And whichever gene is in the middle will be ending up being in the middle. Let's just simplify this. So let's say we have A, B, and C genes. You know my drawing is not the best, right? And then we have X, Y, and Z genes here, right? So when crossover takes place, let me pick a different color here. When crossover takes place, so we have one cross here. Okay, come on. Cross here, and then here, and then the second crossing is taking place here. So we end up having A, Y, C, and X, B, Z, which means whichever gene is in the middle is being flipped. So this is basically more civilized version of what I've just drawn for you on an example that was given previously, where we have 3 genes and we have double crossing, and the one that is in the middle, which is the PR, is being swapped. Just to note here that those are sister chromosomes of the homolog chromosome of F1 females. Here is very simplified visual representation of what we have just talked about when we are having crossover, double crossover, and what does it cause? What is the result? So we already know that the distance between VG and B is 17.7 and VG PR distance is 12.3 and B PR distance is 6.4. So now let's look at the calculation of the gene distances. So if we take under consideration the VGPR distance and BPR distance, then we can calculate the sum and it will be 18.7 MOP unit. However, the VGB distance is 17.7, but as we talked previously, it's more accurate to take under consideration the small fragments and sum it up. That will be more accurate measurement for the gene. There should be an explanation, right? Why we are having differences when we think we have calculated right the VGB distance and also the VGPR plus PRB distance. Why are we getting different numbers? There is an explanation. It's not a magic. There is an explanation to it. And the explanation is that for VGB distance, the double crossover was not taken under consideration. When the double crossover is taken under consideration, even for VGB distance, we would get the correct number, which would be bigger than 17.7. And that is the 18.7, which is the VGB distance by summing up two parts, VGPR plus PRB. Now let's talk about interference and see how the number of double crossovers may be less than expected. So a new terminology, chromosomal interference, that's the occurrence of crossover in one portion of chromosome interferes with crossover in an adjacent part of the chromosome. So to translate it into more human language, that means that if there is occurrence of crossover in one part of the chromosome, that will change the probability of crossover in adjacent part of the chromosome. It can go both ways. It can increase or decrease the probability. And there's coming also non-uniformity between chromosomes or within a chromosome by these chromosomal interference. We can compare observed and expected frequencies of double crossovers and introduce a new measurement, which will be the coefficient of coincidence. That is the ratio of observed double crossover frequency, the ratio of that with expected double crossover frequency, and also the interference would be the measurement that equals to 1 minus the coefficient of the coincidence. So expected probability of double crossover is the product of the single crossover frequencies in each interval. So the probability of the single crossover between VG and PR is 0.123, 12.3 MOP unit, and the probability of the single crossover between PR and B is 0.064, which is 6.4 MOP units. So if the interference is 0, then the crossover between adjacent regions are independent events, and if the interference is 1, then the double crossover takes place. So let's look at the application of what we have talked about. So there is the expected probability of double crossover, which we can calculate based on the numbers that were shown at on the previous slide. So 0.123 times 0.064, which equals to 0.079, which is the 17.79% probability. And then the observed proportion of double crossovers will be 13 plus 9 divided by 4197 times 100 for percent purposes. which is 0.52%. So the coefficient of the coincidence was the ratio, if we remember from 2 slides before. That will be 0.66, and the interference would be 1 minus 0.66, which will be 0.34. So now we can see whatever we were talking about actually has an application, and we can use to calculate some measurements. So what do you think? Does genetic map correlate with physical reality? Uh, I think answer is pretty straightforward. Order of genes revealed by genetic mapping corresponds to the actual order of genes along the chromosomes. However, the actual physical distance does not always show direct correspondence to genetic distance. And there are multiple reasons why that's the case. One of them is double, triple, and more crossovers. We talked about that already, and we have shown that addition of small portions of the map are not always representing the distance of the full distance of the gene. Also, there is a 50% limit on observable recombination frequencies. Another reason is the non-uniform recombination frequency that occurs across chromosomes, and also the mapping functions compensate for some inaccuracies, and then also recombination rates are different between different species. And now, as we already know by now, we don't imagine our life in genetics without Drosophila. And again, there is another example of Drosophila melanogaster that has 4 linkage groups. And it is important to note that when we see linkage crossover, which is kind of synonymous, that kind of explains that these genes are on the same chromosome. So during our next lecture, we are going to talk more about mechanisms of recombination, and we will use the 2 model organisms for understanding those mechanisms, and those would be the Baker's yeast and the bread mold. The more scientific terminology for Baker's yeast is Saccharomyces cerevisiae, and for the bread mold is Neospora crassa. And we are going to use these 2 models to understand more details the mechanism of the recombinations. So all 4 haploid products of each meiosis are contained within an ascus, which is the sac. And then the ascospores, the haplospores, can germinate and survive as viable haploids that divide by mitosis here. And then the tetrad, the 4 ascospore in a single ascus. And then haploid strains of opposite mating type A and alpha can be mated, and the resulting diploid induced to undergo the meiosis. And then we are dealing with the cycle over and over. This results in very interesting genetics, and we will talk about it more detailed during our next class.